Thank you very much for the kind introduction. Good afternoon, ladies and gentlemen. The aim of my talk is to give you an overview of my work doing my PhD and my time as a group leader at the Ludwig Boltzmann Institute for Tissue Regeneration and Traumatology in Vienna. And now I'm working at the University of Leeds on advanced gene-activated matrices for tissue interface regeneration, which I might mention at the end of my talk. So the clinical situation in segmental bone defects, as you all might know, is that you have the problem of a very large tissue defect that does not heal properly. And therefore, you would need a minimally invasive, potentially one-step procedure that can be applied in the operating theater, and this therapy should be cost-effective and safe. Currently, fracture management and large segmental bone defects in the clinic are treated by using autologous bone grafting from the iliac crest, which is the gold standard, and that leads to donor site morbidity. And you can also use recombinant bone morphogenetic proteins, which have to be applied in supraphysiological dosing and might lead to adverse effects. And also a very large field is biomaterials at the moment. We thought that an ideal therapeutic approach replacing all these experimental methods at the moment might be non-viral transient somatic gene therapy. So we basically manipulate cells resident at the defect site with expression constructs to overexpress trophic factors that locally in situ induce the regeneration of the defected tissue. As you all might know, are familiar with gene therapy, I guess. So we're aiming at introducing therapeutic DNA to the patient cells in the body, encoding a growth factor, and by encoding a growth factor, the cells then secrete this growth factor, which lead to the autocrine and paracrine stimuli at the defect site that induce regeneration. The advantage of a gene therapy over a recombinant growth factor therapy is that with a recombinant protein, you always, most of the time, get a very high burst release at the beginning of the therapy that then declines below therapeutic effective levels or is above therapeutic levels at the beginning of the therapy, which leads to adverse effects. With gene therapy, in contrast, you can induce a sustained release by the patient's own cells in the defect site of very low doses of highly bioactive protein, which then gives you a better window of opportunity for therapy. So what are the requirements for non-viral gene therapy? Of course, you need an in vivo transfection method. Sometimes, and I will get into this later, you need to have an effective mode of delivery, like a gene-activated matrix. You need suitable therapeutic genes and a suitable expression vector. So suitable therapeutic genes, we decided for our work to work with bone morphogenetic proteins, which are very potent inductors of bone formation, as you might know. And I'm not going to get into much detail with this because actually I've seen that in the afternoon there's a talk by Dr. Kawai on the delivery of heterodimeric bone morphogenetic proteins, which are very highly bioactive, and that has been pioneered by that group where I was just starting my PhD. But the basic idea is to not only deliver one gene at a time, but to deliver a combination of BMP2 and BMP7 genes, which then, when the cell expresses these genes, leads to the formation of BMP27 heterodimeric growth factors, which have a very high bioactivity compared to the homodimeric growth factors. So we were looking into designing expression vectors, and you will see also later on that Dr. Kawai also pioneered this work. They also devised very suitable expression vectors for this approach. We used the CMV promoter co-expression plasmid for BMP2 and BMP7 genes. This is an FDA guideline-compliant gene therapy vector for vaccination studies, and that leads to constitutive co-expression of bone morphogenetic protein 2 and 7. What we also aim to do is in terms of a temporal control of the gene delivered, so we want to be in control of gene expression, is a TET-on system, which works by induction by doxycycline to activate expression. So we can activate expression via an inducer substance through a tetracycline responsive element in the plasmid. It's a bidirectional plasmid that has two reading frames for bone morphogenetic protein 7 and 2. And what we also did is, usually those are two-component vector systems. You have a response vector and you have an activator vector that constitutively expresses the transactivator that responds to doxycycline and mediates co-expression from the TET response element. And we also introduced this expression cassette into the same vector. So this is a vector that has 3 expression cassettes and can be delivered as one single therapeutic. So the non-viral transfection method, we decided to use ultrasound-mediated gene transfer in vivo because it's a very suitable non-viral gene delivery method. It works with a physical ultrasound stimulus that induces a cell membrane poration at the target site. And we started to work with this because we were interested in the possibility of applying a transcutaneous trigger that can ideally also be focused in space, so we have spatial control of the gene therapy. So how does zone operation work? Basically, you need a neutral ultrasound contrast agent, which are microbubbles. And these microbubbles start oscillating in the ultrasonic field and then induce a cavitation effect that leads to the transient cell membrane poration you need for the cells to take up the DNA. And in our initial studies, we did a study in mice and the ectopic muscle, and we used a constitutive co-expression plasmid for BMP27. And we also had an internal control in the same sample of luciferase that we are able to monitor gene expression in vivo in real time. We then applied an ultrasonic field using the sonoperator device and monitored gene expression and bone formation in the ectopic model. So we had 3 groups. One group was passive gene delivery, where we just injected the nucleic acid therapeutic without any additional stimulus. And then we had 2 zone operation groups, basically with different power settings, 4 watts or 2 watts per square centimeter. We used 20 micrograms of the internal luciferase control and 20 micrograms of the constitutive co-expression plasmid, or 20 micrograms of the inducible co-expression plasmid. We used 4 times 10 to the power of 8 per ml microbubbles in the sample, and then we applied the 1 megahertz ultrasound with these settings. And this is another thing that has been found out by many groups working in this field with non-viral gene delivery of bone morphogenetic proteins. At the moment, efficacy is quite low, so you have to repeat this treatment at least 5 times to get ectopic bone formation, which is an issue we'll get into later on. And what we found basically is by monitoring the luciferase activity is that if you apply zone operation with increasing power, you get increasing gene transfer efficacy. So basically, compared to passive gene delivery, you get a higher percentage of animals that are positive for gene transfer after treatment. Interestingly, in terms of gene expression levels, there was no difference between passive gene delivery and zone operation delivery in our setting. But we were able to increase the success of gene transfer by using ultrasound. Then we used the constitutive co-expression plasmid and had a look at bone formation efficacy. We applied the ultrasound again with that regimen for 5 days. And then we monitored bone formation in the hind limbs of these mice using an in vivo micro-CT. So we had got bone formation data for 2 time points, 14 days and 28 days. And as you can see very nicely here, with using the constitutive system that expresses the therapeutic all the time during the therapy, we get very nice formation of ectopic bone structures. Interestingly, the effect and the extent of bone formation, there was no difference between ultrasound gene transfer and passive gene transfer. We did the same using the doxycycline-inducible plasmid, but for activating expression from that plasmid, we of course had to give doxycycline to these animals. And we decided not to put the doxycycline into the drinking water, but to inject it intraperitoneally to make sure that the animals received a suitable dose of doxycycline, that we actually can see bone formation. And it has been repeated for 7 days. So the expression time window we achieved was for 7 days. And as you can see here, also with the inducible system, you get ectopic bone formation, although to a lower extent compared with the constitutively expressing co-expression system. And that is actually related to the gene dose, because with the inducible system, we only expressed the therapeutic transgenes for 7 days, whereas with the constitutive system, expression was on throughout the experiment. And if you have a look at the bone volumes, you can see that they're pretty much all over the place. So that's what you see in ectopic bone formation sometimes, especially with a relatively low efficacy therapy. But if you have a look at we then went into histology and verified that these ectopic structures were indeed bone. And the interesting thing is, as you can see here, is with this gene therapy, just by applying a gene therapeutic with no cells, no biomaterial, we get the formation of a stem cell niche in the ectopic environment. So you can see these ectopic bone structures have a hematopoietic bone marrow. And this has been confirmed as bone by Francoso staining as well. And we also stained for the internal control luciferase plasmid. So you can see that the cells that are actually forming the ectopic bone are the cells that are expressing the transgene that has been delivered. And if you have a closer look at the hematopoietic bone marrow, you can see that this is indeed hematopoietic bone marrow of the host that is established in these ectopic bone structures that were induced by the gene therapy. If you have a look at the bone formation efficacy, and that's where you can see the difference between passive gene delivery and sonoporation, you can see there's a significant increase in ectopic bone formation efficacy in the constitutive system. And that effect becomes even more apparent if you look at the inducible system, because the gene dose is reduced. And you can see at the lower gene dose, you get only bone formation if you use ultrasound-mediated gene delivery and not by passive sonoporation. And of course, the system without doxycycline is silent, so you get exclusive expression of the therapeutic only if you add the inductor of doxycycline, which gives you a very high degree of control of the therapy. We then decided to move to an autotopic model to try this therapy in a setting that is relevant for the clinic and in a bone fracture. So we had a red femur defect model, which was a 4 millimeter defect that was stabilized by a plate with screws. And then we started basically a similar regimen to the ectopic experiments, where we used the constitutive system this time because we wanted to make sure that this works. And then we applied the treatment for 5 days, starting 3 days after setting the fracture in the animals. Did luciferase imaging for the control if the gene transfer was successful, and then evaluated bone formation with micro-CT 4 weeks and 8 weeks post application of the therapy. Again, we injected the mixture of microbubbles and DNA into the fracture cavity and applied ultrasound using the protocol I described for the ectopic formation model. And as you can see, if you do bioluminescence imaging in the rats, which costs a lot of money because you need a lot of luciferin for a rat compared to a mouse, we get positive gene expression at the ectopic fracture site. And you can see that the autotopic gene transfer efficacy indicates a positive effect of sonoporation, although not significant in this setting. And you can see that the luciferase activity is, interestingly enough, reduced if you use an internal control setting. So if you use a luciferase plasmid and a therapeutic plasmid, you depress overall gene expression, which might be an important thing for people to consider that use luciferase internal controls. It might reduce your therapeutic efficacy. And if we have a look at the targeted gene expression, so the localized spatial control of our delivery, we saw indication that this is confined to the fracture site, and then decided to open the animals and carry out luciferase imaging on the open animal and explants of the femur. And you can see that we were able to deliver our therapeutic specifically to the fracture gap, and that gene expression is pretty much limited to the fracture gap and the target site we intended to apply the therapy to. We then also wanted to track our nucleic acid, because we wanted to see what happens to the DNA if you inject it as a high dose into fracture gap. Where does it go locally? We did not do a systemic biodistribution. And then I came across the work of a group from Switzerland that actually worked on a thymidine analogon, which is basically, if you use like bromodesoxyuridine or other analog, it might like interfere with your expression. But this group actually showed that their analogon FR-EDU is actually not interfering with any biological process. So we decided to label our DNA using a metabolic labeling approach by feeding thymidine oxotrophic E. coli with the analogon during plasmid amplification. So we get this functional group in our plasmid DNA, and that can be then later on very easily be detected in histology using a very specific copper-catalyzed imine click reaction. So we processed our femurs using histology, and then used a near-infrared probe that clicks to the thymidine that has been modified in our plasmid DNA to detect the DNA at the defect site. And using a near-infrared scanner, we were able to demonstrate that actually quite a lot of the DNA that has been administered binds to the intact bone in the fracture, which might be a reason why it's not very effective, because the DNA gets immobilized. And if you think about like purification of DNA, where you use apatite in chromatography and native bone, it might be that DNA binds to native bone in a similar mechanism it binds to apatite columns. And you can see that some of the DNA also goes to the hematopoietic bone marrow. This is the DNA bound to the cortical bone in the intact fracture. And there was very, very little DNA detectable in the surrounding muscular tissue in the femur, around the femur, sorry. Then we had a look at the bone formation efficacy. And basically, we saw an indication that sonoporation increases bone volume in the defect side, although it was not significant at this point in time, and I will get into this problem in just a minute. We had a look at the in vivo micro-CT scans of 2 weeks and 4 weeks. So light blue is the 4 weeks bone formation, dark blue is the 8 weeks bone formation. 0 indicates the defect margins. And you can see here, this is the Sono BMP group, is the group where the BMP plasmid has been applied with sonoporation. The sonoluciferase was a luciferase only control. So basically that's our negative control for the therapy, not containing the therapeutically active plasmid. And then we had a vector control where we just used some random empty plasmid and an empty control group. And you can see that only in the BMP group we got 2 out of 6 animals that actually display the fracture union, which is not like very, very good, but it's a very good indication that what we are trying to do is actually working. So these are the potential unions we found in the treatment group. And it was decided to class this as a not complete union, but we got 2 complete healing of the fractures. And these are the controls. We got one union in the vector control, which might be indicating that the model defect size was not sufficient enough to produce a 100% non-union defect. And we do not get any unions in the empty group or in the sono-look group, which contains the luciferase plasmid. So basically, we get an increase in femur union rate using the sonoporation protocol and the BMP co-expression plasmid, although significance will have to be demonstrated. So the conclusions are that sonoporatic non-viral in vivo gene transfer enables transient transgene expression for up to 28 days and the induction of ectopic bone formation in vivo. That the sonoporation increases the probability of successful gene transfer compared to passive gene transfer in ectopic and autotopic settings. That the inducible gene expression system showed that expression for 7 days is sufficient for causing ectopic bone formation, although ectopic bone formation is reduced compared to the constitutive system that delivers a higher gene dose. And that autotopic sonoporation is a feasible gene transfer modality for in situ gene transfer into bone defects autotopically. The co-transfer of an internal luciferase plasmid, which is very elegant and allows you to see gene expression, can lead to competitive expression from the other plasmid that impairs your therapeutic level, so you need to be aware of that. And that autotopic sonoporation of BMP2 and 7 shows a trend towards enhanced regeneration in critical size defects without cells or biomaterials. So the questions that are still open is like that the plasmid DNA binds to the bone matrix in the defect and thereby maybe reduce therapeutic efficacy. And the gene transfer efficacy, as with most non-viral methods, is still low. So at relatively high doses, so we need to optimize gene delivery. And that brings me to the last part of my talk, which is the last method we developed. And we were thinking about an autotopic setting. The problems might be that, first of all, the DNA might bind to the bone. And then the next problem is you always need to think about, if you do in vivo gene therapies, you need expression-capable cells at the defect site. Because if you deliver DNA to, like, for example, a non-union where there's a lot of fibrotic tissue, the cells that are present there might not produce very high amounts of transgene. So we were thinking of enhancing this approach by using a method we dubbed matrix-assisted sonoporation, which is a combination of a cell therapy and an in vivo gene therapy in a novel gene-activated matrix approach. So gene-activated matrices, as you all might know, are biomaterials that non-virally deliver genes to a wound healing site. They are biocompatible. The DNA uptake is mediated by invading host progenitor cells, and there have been a lot of systems developed using several different kind of transfection reagents, and basically it leads to transient sustained delivery of growth factor genes again, and this transient sustained release is mediated by cells present at the defect site. And we wanted to combine this approach with the approach I showed you before to develop a novel in vivo gene delivery platform. So the aim was the enhancement of sonoporation, because if you think about sonoporation, you need a contrast agent, you need your nucleic acid, you need your target cells, and they all need to be present in the same space in close proximity for the method to work effectively. So we thought if we incorporate this into a biocompatible matrix and enable son-operative gene delivery in a biocompatible matrix where we put in precursor cells along the microbubbles in the DNA, we might improve therapeutic efficacy and we might improve gene delivery efficacy. So basically, again, we put in target cells, plasmid DNA, microbubble contrast reagent, and we use the fibrin collagen hybrid matrix for this approach. This is the matrix here. You can see the cells and the microbubbles and the matrix. Of course, you cannot see the DNA in light microscopy. And we initially carried out experiments in vitro using luciferase as a reporter for gene transfer. And what we found out, and this is basically the take-home message for matrix-assisted son operation, is that this process does not work unless you include collagen into the fibrin matrix. So it does not work in the fibrin matrix alone. It only works in the combination, and of course it only works if you then use ultrasounds and microbubbles. So basically what we have now is a advanced therapeutic construct that you can activate with an external physical stimulus, which is ultrasound. We showed that the gene transfer efficacy in these matrices is of course dependent on applied ultrasound power, and that cell viability is basically not much reduced in a 3D setting, which was also an interesting finding. And then we decided to move on and try the whole approach in vitro using our constitutive co-expression system. And what you could see here is alkaline phosphatase, which is a marker gene for osteogenic differentiation. We used C2, C12 cells, which are an in vitro model system for osteogenic transdifferentiation. And you can see that by using the matrix-assisted son operation approach, we can produce a significantly enhanced differentiation stimulus compared to matrices that have either no ultrasound treatment or no microbubble contrast reagent. So again, this is a son operation process that is going on in these matrices. We had a look at the histological appearance of these matrices after 14 days of differentiation in vitro. And you could see here in the positive control where we used recombinant BMP as a control. The top is Fancosa staining, so everything black are mineral deposits. Red is the nuclear counter staining, and the bottom here is osteocalcin staining for osteocalcin, which is a specific marker for osteogenic differentiation. And you can see that in the recombinant control, indeed, we get very robust osteogenic differentiation. There was no differentiation in the negative control that did not contain any therapeutic DNA or growth factor. And then if you have a look at the samples that were treated with matrix-assisted son operation, I don't know if you can see it, but I have a higher magnification image as well. You can see a lot of mineral deposits forming in the matrix after in vitro differentiation and positive osteocalcin staining. In the sample that has not been treated with ultrasound but contains the therapeutic DNA, you can see a little bit of osteogenic differentiation, but not very much. And the same is true if you do not add microbubbles. So again, differentiation is specifically dependent on matrix-assisted son operation. These are the matrices in a bit more detail, so you can actually see the mineral deposits. And then we decided, okay, this was very encouraging. Let's go into an in vivo setting, and we decided to use the system in an ectopic model in vivo. So we, again, injected all the precursors for the matrix formation into the hind limb muscles of mice. And the advantage is all of this is a liquid system, but then once you inject it, because the fibrin and the collagen, it solidifies in the defect setting. So it's a minimally invasive approach again. And then we had a look at a matrix-assisted son operation versus passive gene delivery. And as you can see here, we get very robust ectopic bone formation again in the matrix-assisted son operation group. And I forgot to mention the cell type we were using were not stem cells. In this setting, we used the C2, C12 cells in an immunodeficient nude mouse model as an initial characterization. So this is not stem cell therapy yet. This is the control sample where there was no ultrasound used and there were not that many animals that were positive. Again, this is the comparison between passive gene transfer and matrix-assisted son operation. And then we had a control group where we did not use cells. So this again is maybe reflecting back to the first study I showed, which is just ultrasound gene delivery. And you can compare these methods now directly. And you can see that this one is more effective. And this was a control for the auto-differentiation of the cells, because every time you implant cells, you also need to control what are the cells doing if they're not being treated. And in one animal, we found very little evidence of very small bone formation. Do I still have time? Or? Okay, I'm very sorry. This is the last thing I wanted to show you. With the matrix-assisted sonoporation, we were actually able to generate an ectopic structure that does not only contain bone and hematopoietic bone marrow, but also a cartilage interface. And this is maybe an indication that we can induce complex tissue interfaces as well by using this gene therapy. Sorry for running over time. Thank you. In the in vivo setting? Yes. Yeah, sorry, I didn't include that. Usually you don't see any bone formation if you implant it ectopically. In the bone setting, we have work on fibrin as a bone regeneration matrix. That would be a very important control because in an autotopic setting, fibrin can also induce regeneration, although to a very limited extent, yes.
But over time, it might be, I mean, it might have some efficacy as well, right?
If you do an autotopic study, you will have to include that control, definitely, yeah.
Can you not induce the growth factors into the matrix, the last models that we're doing, without having to have gene expression? In other words, can't you just use the matrix to lock the growth factors in the location where you're looking for growth?
The recombinant growth factor, you mean? Yeah, of course, that has been done quite a lot, actually, with different kinds of biomaterials. They also produce a sustained release and not only a burst release over time. But the interesting thing is with BMPs, if you apply them recombinantly, you still have to use them at very high doses. Whereas the doses we produce with the gene therapy are in the picogram range.
How often does this factor union fail if you use BMPs?
If you use BMP, a recombinant BMP.
How often does it fail without any, just putting it to the other, then with BMPs, so that I can understand how important this question is?
Well, I mean, I'm not a trauma surgeon, but as I understand, I spoke to a lot of trauma surgeons at our institute, and generally, in the clinic, if it's not a very large defect and maybe the patient is compromised as well by infection, so it's not that much of a problem. It's maybe 30% depending on the fracture site. But then once this chronic condition is established, it actually puts a very high burden on the patient because then it is very difficult to regenerate it even with traditional methods as far as I know.
So you're not talking about primary healing, but you're talking about a non-union?
Yeah, I'm talking about non-union defects, large size defects, yes. Yeah, you will have to debride the defect site because you will have fibrous tissue present there, which might not be conducive to healing. That's correct.
And can you put just that cadaveric bone in that site to be able to make the union occur?
Well, there has been work done in that area where they use and decellularization as well as a big topic at the moment. But then again, with cadaveric bone, there has been a lot of work absorbing recombinant BMPs onto it and using it as a scaffold. But then again, you have a problem with you need to test it for the presence of pathogens. It's a primary material coming from other human patients. So there are ethical implications and cost implications as well, of course.
Okay, thank you so much.
Thank you very much.